Hi everyone, welcome back to my A-level biology lecture series. In this lesson, we will explore Huntington disease. At the end of this lesson, you will be able to explain the relationship between genes, proteins, and phenotypes with respect to the Huntington gene, HTT, Huntington protein, and Huntington disease. Let's get started. Huntington disease is an autosomal dominant neurodegenerative disorder caused by progressive loss of neurons, particularly in the brain basal ganglia and cerebral cortex. So what are the symptoms? So the symptoms include motor disturbance, which includes uncoordinated jerky movement, progressive physical disability, particularly increasing difficulty with voluntary movement, leading to the loss of speech, and cognitive decline, where memory loss, impaired judgment, and eventual dementia will occur. Symptoms most commonly develop between the age of 30 to 50. Basically, this is an adult onset disorder, although in some cases they may appear early, or we call it as juvenile onset or later in life. And this is particularly important to take notes because at 30 to 50 years, an individual may already develop or have his or her family. And Huntington disease is an autosomal dominant disorder. So therefore, the child may inherit a copy of this mutated allele or the dominant allele, and the child will develop the disease. So Huntington disease results from the mutation in at least one copy of the Huntington gene, which is located on chromosome 4. The mutation follow an autosomal dominant pattern of inheritance. So basically means a single defective allele is sufficient to cause the disease. The Huntington protein normally interact with at least 19 other proteins, 6 involved in transcription, 4 involved in intracellular transport, 3 involved in cell signaling, and the remaining 6 where their functions remain unknown. So generally, Huntington protein are quite important to regulate the cellular activities. Huntington disease is caused by an expansion of the CAG trinucleotide repeat in the coding regions of the Huntington gene. So this CAG triplet specifies the amino acid glutamine. So therefore, the expansion leads to an abnormally long polyglutamine track in the Huntington protein. So you can see that CAG continuously occurs in the DNA or Huntington gene, results in the glutamine or polyglutamine chain. So increasing the number of CAG repeats is associated with genetic anticipation. So genetic anticipation means the tendency for symptom to appear at the younger age and progress more rapidly in the successive generation. So normally, a normal individual will have about 10 to 35 repeats, produce Huntington protein with a normal polyglutamine thread, allow normal functions of the Huntington protein. However, in Huntington disease, this Huntington gene mutation occurs with 36 to more than 120 repeats. And this leads to the synthesis of mutant Huntington protein containing an abnormally long polyglutamine thread, which disrupt the normal protein folding as well as the functions of Huntington protein. So a person with 36 to 39 repeat may experience reduced penetrance, This individual may or may not develop clinical symptoms during their lifetime. However, if symptoms do appear, the onset is typically later and the severity is milder compared to the case with the full penetrance. So what is full penetrance? Full penetrance means the individual with more than 40 repeats and symptoms almost always occur and typically with early onset and greater severity. What is the pathologies at the cellular level? So first, we have the post-translational cleavage of mutant Huntington protein, which generate fragment containing an abnormally long polyglutamine thread. So the excess glutamine residue increase the likelihood of abnormal protein-protein interaction. And this fragment bind together through the hydrogen bond, forming insoluble protein aggregates instead of correctly folded into the functional protein. And over time, aggregate accumulates within neuron, disrupting normal cellular processes and causing neural dysfunction. So these actually explain why the disease is adult onset, because it takes time to accumulate sufficient aggregates to disrupt the normal cellular process within the neuron. So misfolded fragment can join or form these inclusion bodies within the nucleus or cytoplasm of neurons, further impairing their functions and survival. So in this diagram, you can see that on the left-hand side, we have the normal Huntington gene and also the Huntington gene with CAG repeats. So one copy of allele is sufficient to trigger the disease, because with one copy of allele with CAG repeat, it will form this protein aggregate, which disrupt the normal functions of the neurons. And with that, we have come to the end of this lesson. Hope you found it helpful and easy to follow. If you have any question, feel free to drop them in the comment section. See you in the next lesson. Have a great day.